Just a minute, just a minute. Okay, good afternoon to everyone. Prashant, thank you so much for such a nice introduction. Myself, Sivaramaiah Nallapeta, you can call me Shiv, which is short and simple. I'm heading this business operations for Nanotemper Technologies based in Bangalore. So our company is pioneer in providing some solutions for the protein characterization. So I'll give you a quick overview about the technologies and some applications which the people working in this protein biology might find interesting. So please feel free to stop me anywhere if you have any questions. So before going into the products, technology, and all, I just want to give you small overview of the historic introduction, how this company has came in. So the two CEOs of our company during their PhD, they filed a patent. They established that when thermophoresis is done in a very small scale, that can be used for the biomolecular interactions. With that, the instrument monolith NT115 came into the picture, and later on several versions, several developments on this technology happened. And other than this, the Nanotemper also provides a technology for the protein stability. So at what temperature your protein is stable and how the colloidal stability is important in the characterization. So all this kind of different platforms are available with the Nanotemper. And most importantly, we also have a Tycho, a very small instrument which tells about the protein quality in just 3 minutes. So all this quick portfolio, I will just give you a small introduction with the basic principle behind it and some applications which you might find really interesting. So these are our two CEOs, the Stefan and Philip, who started this company, to be honest, like in a small incubation center in the university. And now we are very proud and glad to say that, it has become one of the fast growing company, which has globally spread across in different countries. And the company we started with 2 people now have more than 250 people, and we have more than 1,000 installations. And most importantly, this company is connected most scientifically with the customers, the publications. We have 4,000 publications where these technologies have been acknowledged and used in the scientific workflows. So with this quick introduction, now I quickly take you into the details. So normally, when anybody working with the protein, so they need to know what kind of a protein or what quality of the protein they are working. Else, you don't get any useful information out of it. So let us see some few interesting slides. You see here, there are some vials where there is a protein in this. So can you visually tell in which vial the protein is stable or the protein is functional? Of course not. Nobody can tell that. And one other thing is, in our normal research, we always store our proteins in the freezer. Like maybe once the experiment is over, you was just throwing it in a freezer, and after some time you are getting it back by thawing it and then using the protein sample. But are you sure, when you are freeze thawing it, how much percentage of the protein is losing its functionality? Are we able to assess any of these things? So technically speaking, there are so many tools to understand the protein characterization. One is for suppose you want to know the concentration, you just go to the spectrophotometer and just see the ratio. And if you want to know the size and molecular weight, you run a simple STS page. And if you have a mixture, then you go for many separation techniques like chromatography and other things. But in none of these techniques, you are not able to tell whether your protein is a functional or not, whether the protein is in a good quality or not. So that's where the company felt that there should be a platform to assess the quality of the protein. So what they made here is, by measuring the intrinsic tryptophan fluorescence, you can evaluate the quality of the protein. So normally, when the protein is folded, all the tryptophans are buried inside. So as in how you are heating it up, the tryptophans gets exposed, and the change in the fluorescence will contribute to the stability of your protein. So in that way, we can tell whether your protein is folded or unfolded. If it is completely opened up, there is no function, and if it is folded, then it is functional. So they devised a small instrument, Tycho. This is the footprint of the instrument, where you have an option of putting 6 different samples. All it does, it heats the sample from 35 to 90 degrees in 3 minutes and just measure the intrinsic tryptophan fluorescence. So with such a simple platform, we can do a multiple applications. Anybody working with the proteins can understand whether your protein is present in the sample or not, whether it is pure or not, whether in a particular protein purification, when you are purifying the same protein, you want to know the quality at the end of the day. The protein which was purified yesterday and protein which was purified after 10 days, is the same quality of the protein is retained or not. And other thing is the functionality. So we all know that if the protein is not functional, then none of the bioassays or make sense. So before going into that, you should always know whether my protein is functional or not. Unless the protein is functional, you cannot do any tests. So to check the functionality, and also now you can also tell, like a spectrophotometer can tell the concentration of the protein present in the sample. So this small instrument, in 3 minutes of time with the 10 microliters of the sample, it will give you the multiple parameters to study the quality of your protein. As I said, this can be very much integrated in any protein workflow, right from protein purification, chromatography. Like if you are eluting the different chromatographic fractions, you always want to know where is your target protein. So in that way, instead of running a simple SDS, you can just take this in a few microliters, and without any sample processing, you can know in which fraction your protein of interest is there. So, and also to study the purity of your protein and storage. As I said, like when you are storing the proteins for the longer time, you always need to know when you are using it for the next time whether it is functional or not. And assay development, this is very critical in the biophysics. So if you're doing any bioassay, if you're doing any a method to study the proteins, then obviously the quality is very crucial. In this way, these instruments help you to assess the protein quality in different workflows. So one other technology what Nanotemper is famous for is microscale thermophoresis, which is a short form known as an MST, which is a very popular word in this biophysical world. So normally in the microscale thermophoresis is a biophysical method that measures the interaction between the any biomolecules. What it does is it just measures the fluorescence change, which is a result of the IR laser inducing the temperature. So normally there are two important things that contributes to the MST signal. One is thermophoresis. Thermophoresis, the name itself will define that phoresis is the movement, thermo is the temperature, the motion, the movement of the molecules in the presence of a heat is called a thermophoresis. But this thermophoresis is dependent on three important things. One is the size, charge, and the conformation or hydration shell. What it says is whenever the two biomolecules are interacting, any one of this parameter like the size or a charge or the hydration shell is changed to happen. When there is a small change in any of these parameters, the thermophoresis signal is also measurable and quantifiable. In this way, we can always measure the biomolecular interactions by the quantifying the thermophoresis. And other important thing is the trick. Trick is as I said, we are working with the fluorescence, as we are working with the fluorophore, and the temperature is involved here. The fluorescence is always dependent on the temperature change, and the temperature is also dependent upon the changes in the local environment of the dye, what it is present. So even the binding of the ligand is strongly influenced by the change in fluorescence. So normally when you are studying the biomolecular interactions through thermophoresis, either trick or thermophoresis is used to measure. So now let us see what is the basic setup of the instrument. The instrument is pretty simple. It is not even half a meter of bench space is required for it. And if you look into the inside system, we have two important components. As I said, the fluorescence is involved. So to read out the fluorescence, you have an LED excitation and the fluorescent detection cubes. And the second thing is a IR laser. IR laser is very important to create the temperature so that the molecules will move because of the heat, and that is measured what you quantified as a thermophoresis. So in principle, what we do is you label one other partner and titrate the ligand of the different concentrations. And further, you take them into a small glass capillaries and then subject every glass capillary with a different concentration of a ligand for the thermophoresis by the laser. So normally what happens, there is a fluorophore and there is a ligand also is there. The moment you put some heat on it, the molecules will move away from the heat. Then obviously the fluorescence is changed. That change in fluorescence is actually recorded in all the different concentrations of the ligand what you have titrated it. And the most important thing here is you are not immobilizing. You are directly taking in the native conditions of any buffer. and all you require is 4 microliters for the sample measurement. And if you look into the dynamic range, how sensitive is this instrument, it can measure even the picomolar affinities. And of course, the higher will go for even the millimolar range also. And the most importantly, the time taken for the measurement is all about like 10 minutes of time. So in 10 minutes of time, you prepare your dilutions, put them into the glass, load into the glass capillaries and put in the instrument. It will record the thermophoresis for all the different concentrations. So typically how it looks is like, for every concentration, the thermophoresis is measured. As you can see here, initially you record the fluorescence, and after that you put the laser on. The moment you put the laser on, there is a heat. So because of the heat, the fluorescence is going down, and this is taken for 30 seconds. And after that, you put the laser off, then the fluorescence is regained. But what is happening in all these traces is like, at one particular concentration, your ligand and the protein, or your ligand and the other binding partner, or bind to each other. When they bind, then the movement will not be the same. Then, they move apart from each other. This differences is good enough to tell at what concentration protein is binding to your target or a ligand. So further, we have a specific software like, what we call it, the KD fits, or like a sigmoidal curves, where it will translate the thermophoresis data into a fit. So here you can see here at the low concentrations, like we call it as like, a baseline or an unbound state. And here you see they are getting saturated. There is no any increase in the there is no any increase. And generally the midpoint is taken as a binding affinity. So in this way, just by labeling one of your partner and titrating the ligand in 10 minutes of time, you can determine the KD value without any challenges. This is the biggest advantage what we have in this technique. So hope everybody is clear like how this the principle and everything works. So this is again the footprint of that instrument, and if you can see the major what are the important features what this instrument can do is it can measure the KD independent of size and mass of binding partners. The biggest advantage here is, since we are measuring the movement, since we are measuring the thermophoresis, there is no any limitation with the size. Any binding, it can be like an ionic binding or a small molecule, or a a bigger protein complex, or even the ribosomal protein complex. Anything can be measured using this instrument. And importantly, you are not immobilizing anything, you are measuring in a solution. Measuring in a solution is directly in a native conditions. You are trying to not change the conformation of any of your partners. So this is a very crucial when you are studying the molecular interactions. And importantly, you don't have any buffer effects, because you are taking only few microliters for the sample measurement. Like even if it is in detergents, for the membrane proteins, everything, this technique works beautifully well. And other than this, as I said, the dynamic range, it can go even for the picomolar affinities. And importantly, we are also able to measure even without a complete purification. Even if it is in a mixture, like cell lysates can also be used for the molecular interactions. Maybe I'll show some quick case studies which you might find it really interesting how this technology is used. And if you look into this, this technology can be compared to the very much to the surface plasmon resonance and ITC. What we mean to say here is the end result in all these techniques is the same. So we have one case study where a protein and DNA interactions were measured, and you can see here in all these three techniques, the binding affinity, the N value is almost the same, like 29 nanomolar, 24 nanomolar, and 30 nanomolar. But why MST is important, but why MST is more successful, or why people prefer this? The more important thing here is, as I said, we work with a very, very small concentration of the samples. We require 10 to 20 nanomolar of the protein for doing the assay. And importantly, the cost. There is no any running cost here. It is just only the cost of your protein, and then further you're doing a titrations and doing the measurement. So this is an advantage in this technique when compared to the other techniques what are available now. So the time, the cost saving, and the speed without any limitations of the sizes what we have. So what all this MST instrument can do? So a competitive assays, maybe I have one very good example to share it with you how the competitive assays are used in life-saving diseases. And you do a lot of screening, like a drug discovery programs, like where you want to know just they are a quick yes or no binding studies, oligomerization, dimer formations and quaternary complex formations. All these kind of studies can also be studied. And importantly, stoichiometry, it will tell you the number of binding sites. It's not just that KD, it also tell you how many possible binding sites are there in the interacting partners. And other things is thermodynamics, like enthalpy. All this information can be derived. For any person working in the biophysics will completely have a complete information about the binding, stoichiometry, thermodynamics while using this instrument. So I will just quickly run through some examples, which I'm sure you'll be finding it very interesting here. So one is with this hepatitis C virus, how the inhibitor binds to the viral protein and then reduces the RNA binding affinity. As we are all aware that NS5 protein is actually involved in the gene replication of the HCV. So when this particular viral protein, binds to the replication factor, there's a very tight affinity of 50 nanomolar. But the same protein, when there are some inhibitor compounds, then you see the affinity is reduced to 94 nanomolar. So in this way, you can, understand how the virulence by the replication has been done by using this MST instrument. So another important thing is the HIV. We all know how lethal is this particular virus. So here normally in the in this virus, if you see the CDR protein, which is a receptor, this is very important for the virus to enter into the cell. When particular the CD4 is glycosylated, then it acts as a shield, and then it doesn't allow the virus to enter inside. So even that kind of studies in the presence of a wild type where there is no glycosylation, and when in the presence of two glycans, you see the affinity has dramatically changed. So like this, all this kind of glycosylation and then the important receptor binding studies can be studied using this particular platform. And importantly, the dimerization. So normally when the monomer, when a protein forms the when the protein binds, then there is a possibility it forms a dimer. So studying these dimers is very important and crucial in the complete cellular process and all the important signaling pathways. So here what we do is normally you take a monomer and other monomer is titrated as a ligand. So whenever the two monomers start binding to themselves forms a dimer, there is a change in the binding affinity. So this changing in the binding affinity is calculated by the thermophoresis. The biggest challenge in this dimers is the sizes are too small, like the techniques, normally the traditional techniques like DLS and SPR, because of such a small size, and since they are dependent on the what do you call this optical based, it's always very challenging, and this can be very nicely addressed using this MST. And another one is here a very good case study like HSP. We all know HSP, heat shock proteins, or few megadaltons. Whenever a small molecule binds to it, the overall the size doesn't affect at all, because something in 10 20 daltons, and then you have some mega dalton protein, the size is absolutely not affected. But here one water molecule is displaced when this small molecule is binding to this protein or HSP. So that small change is good enough for the thermophoresis to determine the binding affinity. So this showcases that this instrument is very sensitive even for a small molecular changes, which is challenging with other platforms traditionally. And I said one more important application of this instrument is even without a complete purification, if your target protein is in the cell lysate, and if you have a histag, or if you have any fluorescently labeled fluorophore attached to your target protein, and when it is expressed, then you can directly titrate your target, maybe it can be a drug compounds or something, and then determine the binding affinity. So in this way you can work both with the purified and not completely purified protein targets when you are using for the MST. And if you just come to the software part, it is very intuitive, it is very simple, and we have a lot of algorithms which is like artificially intelligent based. So once the assay is over, it will tell you whether the data you got is right or not, and if you don't get the data, it will give you some suggestions. Because of this possible reasons, you don't have this data fit properly, so you please go and repeat it. So in this way, in a very simplest way, you can study all kind of molecular interactions without having a simple hurdles here. And the next important topic is aspect is studying the stability of a protein. So the moment we say stability, there are a complicated matrix of attributes. Like you measure the self interaction also as a part of the stability. You do the colloidal stability, that is by the aggregation measurement, and a typical thermal stability, cross interactions, hydrophobicity, and chemical stability. So there are a different attributes to measure the chemical stability, and all of them involves the different techniques and different limitations and different advantages when you are measuring particulars of these things. So normally anybody like working in the protein stability, they require is how you address the stability in a much better way, that is like specificity. So specificity, like for suppose you have a signal from the target and from the excipients or from the buffer components. So it has to be very specific to give a signal to the target what you are analyzing. Of course, resolution in any instrument is important to separate the complex mixtures. And not only just the complex mixture, I mean to say, normally in a protein stability, you have a multiple domains. So in the same protein, you have a multiple domains. So they should be able to separate them. They should be able to separate that multiple domains which might be very closer. And the precision, accuracy, sensitivity, these are all important. When you're talking about any technology, we always go for what is the limit of detection, what is the sensitivity range, how accurate it can give the data. So to address all the stability parameters, we have one another platform which we call it as a Prometheus Panta. Now let us see how this instrument works and how this is useful in the biologics and other biopharmaceutical companies in a various workflows. So we say the principle is nano DSF. It is very similar what we have used in the Tycho in the beginning. So it also measures the intrinsic tryptophan fluorescence. So here you can see when the protein is folded, all the tryptophans are buried inside. When you are heating it up, the protein is opening it up and then the tryptophans are getting exposed. That change in fluorescence is contributing to the stability of your protein. So in this way, you just get a sigmoidal fit, where again the midpoint, where half folded and half unfolded, will contribute to the thermal stability of your particular protein. So in this way, this instrument, by heating it up, we are able to record, tell the TM of your particular proteins what you have. Now, if you look into the basic setup of the same instrument also, you have a simple a tray where you can keep 48 different samples. These are the glass capillaries. Like every sample, every capillary can accommodate one sample. So in principle, you have 48 different samples you are measuring at a time. So what all does is it has the same thing, you have a UV fluorescence where you are exciting the tryptophan fluorescence and collecting the spectra at 330 and 350, very specific to the red and blue shapes. And now in the same instrument, we also have got one more detector, DLS. What DLS actually does is it measures the size of the aggregates, which is very important, which will tell you about the colloidal stability of your particular protein. So in this way, you are recording the thermal stability, you are recording the aggregation and the size of the aggregation in the same sample. All you need is just the 10 microliters of the sample to be filled into the glass capillary. So in principle, 48 different samples, you are measuring the TM, DLS, and the aggregation profile. So now if you look into this, as I said, for the determining the TM, it measures the intrinsic tryptophan fluorescence. And if you want to study at what temperature this protein is getting aggregated, here we have one back reflection optics. It is pretty simple. As long as there is no any aggregation, the light which is shined on it will come back, and that will be measured by the detector. So normally if there are a lot of aggregates, the light gets more scattered, and obviously the back reflection rate will be very less. In this way, you are able to tell at what temperature your protein is getting aggregated. This is very important, like, particularly in the formulations, particularly the proteins, when you are stirring in a different pH conditions, they might get aggregate and then lose the stability. So to know at what temperature and what conditions and what buffer conditions the protein can remain stable, this will be a very important thing. And as I said, like, this instrument also has a DLS. The DLS, normally what we do is, like, you shine a laser and it is scattered from the particles in the solution. And this is, like, a backscattered. And the backscattered is like, nothing but the Brownian motion of the particles. So in this way, you are just measuring the backscattered light and telling the size of your aggregates. So this instrument is more sensitive and it can even detect the size of zero point 0.5 nanometers in the hydrodynamic radius. So just by the Brownian motion of the light which you have scattered in the particles of a solution, you are able to determine the size. Further, what all we do is, like, the fluctuations because of this scattered light are regarded as a function of a time to determine the autocorrelation function. And this autocorrelation function will determine the diffusion coefficient. We have a very strong mathematical algorithms to determine the particle size, and also, like, to determine the diffusion coefficient. In this way, you are determining the TM, you are determining the aggregation, you are determining the size of the aggregates, and also you are telling the polydispersity. Like, is it a homogeneous population, or it is a mixture of different populations, can also be measured in the same run. So this is just like, a screenshot what are all the different parameters you measure and what all you get at the end of the experiment. So basically, companies and many people who are working in the formulations, they want to know in what, in one particular buffer conditions your protein is stable, and how the excipients, how the different buffer composition is playing the stability as an important, can all be measured using this platform. So it can be a very good technology to integrate in the biologics development, and particularly in the long-term stability of your biologics. So you are storing your protein for a longer time and you want to check the stability, how many days, how many years this particular formulation is stable. So all this kind of measurements can be done. And we have more the technical information, which I really thought unless you work on all these things, like, we have a time control, we have a chem control softwares which will tell you a lot of different information in the characterization workflow. So this is all a simple information I want to give it to you. So if you have any questions, please let me know. If anybody working in the protein related workflow, we would be glad to get in touch. And we are very much open, like, doing some proof of concept studies and make the people to understand how these technologies can actually be integrated to your workflow. So we are very proud and glad that, globally we are associated with the the bigger pharmaceutical companies who are using our technologies in their workflow. Like, you can name any biopharmaceutical company, we are already there. And in India, we started 5 years ago, and our journey was more or less similar. Like, when we were, it was a very new technology, and now we are very glad that, we have more than 30 instruments and 60 publications. As I said, that we believe in a lot of a collaborative work. We always help them in writing papers, we always help them with the supporting data what we got it. So that's how we have developed our relations in terms of both collaboration, and in terms of the strength, we have a dedicated teams across the country separately for application and services to support all our customers. And we are very much open for doing a proof of concept studies. Please let me know if any of you are interested in studying the proteins or any of these workflows. We shall be glad to discuss and provide more information to you. And finally, again, I wish to thanks Bioclues for giving me this opportunity, and Prashant for always giving me an opportunity in all the events whenever we are here. Thank you so much, Prashant. So now it's we can have any questions. Please feel free to you can ask me anything. Right, right. Hello, sir. Right, Prashant. Right. Absolutely, Prashant. Absolutely. We have a plans of our business expansion in Sri Lanka and other countries. So maybe I'll get in touch with the professor and then try to organize some event there. Thank you, Prashant. Thank you so much. Sure. Sure, sure, Prashant. Right. What happens to? Yes. Yeah, Prashant, actually, we measure only the aromatic amino acids like tryptophan, tyrosine, and phenylalanine. So the detector is very specific. It only monitors the tryptophan or tyrosine fluorescence, not any other amino acids. Because this, what I said, these amino acids usually, the aromatic acids decides the, the unfolding and folding of this protein. So we monitor specifically only these amino acids. Yeah, like it's all depends on the overall charge. Like when you look into the overall charge of your protein, it depends like what amino acids are actually exposed or how many number of amino acid components are actually deciphering the charge to the protein. So it's slightly different here. We are only talking about the folded, unfolded. So when it is folded, we presume all the tryptophans are inside, and when it is opened up, the tryptophans are exposed. Once it is exposed, that means the protein is opened up. Now it's like denatured kind of. So that time you are only trying to tell at what temperature your protein is opening up. That is the stability of your protein. So that is what exactly we are trying to measure here. Thank you.